The overall goal of this procedure is to isolate the retinal vascular network. This is accomplished by first performing a series of water washes on the isolated retina. In the second step, the retina is digested in trypsin at 37 degrees Celsius for about 1 to 1.5 hours depending on the size of the retina. Next, the vasculature is isolated via additional water washes and dissection under a microscope. Ultimately, pathologic vascular changes such as microaneurysms, capillary degeneration, and abnormal endothelial cell-to-pericyte ratio can be visualized.
So the main advantage of trypsin digest over existing methods like preparing retinal whole flat mounts is that the entire vascular network can be highlighted in great clarity.
Generally, individuals new to this method will struggle because it's technically challenging and difficult to perform consistently. Visual demonstration of this method is critical as the vascular isolation steps are difficult to learn by words alone.
This is a very valuable tool in any laboratory that's performing retinal vascular biology studies, such as in diseases such as diabetic retinopathy. Demonstrating this technique will be Jonathan Chou, a talented medical student from my laboratory.
After fixing the enucleated mouse eye in 10% neutral buffered formalin for 24 hours, place the eye in a petri dish and cover it with a small amount of water. Begin the dissection by using a pair of dissection scissors to carefully make an initial cut in the cornea. Then, while holding the cut area with straight forceps, use a pair of scissors to cut along the border of the cornea and the sclera, removing the cornea.